So today I'll talk about ancestral recombination graphs, which I think are currently revolutionizing how we are doing inferences in population genetics. But I'm going to take a little bit of a step back in my talk about ancestral recombination graphs and talk a little bit about the history of the field. So many of you are familiar with coalescence trees. So a coalescence tree is a tree-based representation of the genetic relationships among individuals in a population in a specific locus, for example, mitochondrial DNA. So if we look from the past to the present and track the history of a population, take a sample and then look at the history particularly of that sample illustrated here in the magenta colored nodes and edges here, we can then extract a tree from that. And that tree is a coalescence tree. And something that happened in the late 80s, early 90s is that people realized that the key to understanding population genetic variation and to make inferences about population level processes, natural selection, demographic history, and so on, the key to that came through considerations of the coalescent tree. And so lots of work proceeded on using coalescence tree for inferences. And perhaps most famously is this Felsenstein equation. And I'm not going to throw too many equations at you, but I think this is a very famous equation that's not being appreciated enough. So I'm just going to advertise a little bit for this equation because it sort of illustrates the way you use gene trees and coalescence trees to make inferences in population genetics. So a little bit about the notation here. X here is the data, and theta, that's the parameters to estimate. So this here we have to the left-hand side of the equation, that's a likelihood function. If we can get that, then we have all of the information about these parameters of interest. So often in statistics, what we're after is we want to be able to see, can we calculate this? The left-hand side of the equation here, the probability of the data, given the parameters. If we can calculate that, then it's sort of the standard tools for then how we would proceed to make parameter estimates, confidence intervals, hypothesis testing, and so on. So to be able to calculate that in population genetics, though, we need to access this coalescence tree of the genealogy. So that's capital G here. So that's that whole tree. And what this equation says is that we can do it by combining two things that we can calculate. The likelihood function from phylogenetics, that's probability of the data given the tree. So that is if you do, say, maximum likelihood estimation of phylogenies or Bayesian inference of phylogenies. That's the likelihood function that you're using there, the way to calculate the probability of the DNA sequencing data given a tree. With this other term here, which is the probability of the tree from coalescence theory. So that's the way that we relate our population genetic models to the data. It's by saying that the population genetic models, the models of history of the population and selection and so on, that induces a probability distribution of coalescence trees. And we can relate that to the data through this function. And then what we need to do is do this integral here. That means that we need to look at all possible coalescence trees and take the product of these two functions for all possible coalescence trees, all possible topologies of them, all possible branch lengths. If we can do that, we can calculate this function. And a lot of work in the 90s focused then on how to do this, how to get at this likelihood function so we could make inferences. A lot of things came out of that, particularly analyzing mitochondrial DNA and Y chromosome. And so here's an example of that. This is from 2015, but it's basically methods that essentially were developed in the late 90s, early 2000s for estimating changes in population size in a population. So the rate at which these lineages merge, what we call coalescences, the rate at which that happens depends on the population size. So if you have a Y chromosome tree like this, or an mtDNA tree like this, you can estimate what the past population size has been. And this is done here in a paper from 2015 for different European different human populations, Europeans, people from the Andes, people from Siberia, and so on. And it's done for Y chromosome and mtDNA independently. And the important thing here is, of course, that mtDNA, that reflects maternal inheritance, and Y chromosome DNA reflects paternal inheritance. And the odd pattern that is in the data, and is still visible in the data, is that while there's been, if you look at mitochondrial DNA, so the female effective population size, We can see that's mostly increased through time. There's a bit of uncertainty. There's some biases here, I think, that causes this not to sort of increase more in very, very recent time. But if you look at the Y chromosome data, there's this very odd bottleneck here that you see in many different populations. And we thought originally, okay, maybe this is selection. There's been sort of a selective sweep, but it happens in many different lineages at about the same time. So not really associated with any particular mutation in this tree. And it's a bit of a conundrum still today, what might cause this. And many people think it has to do with cultural changes, where you get a different social structure, where you get many places in the world, in a relatively short time period, you get a social structure where one male can have many offspring. And most males maybe leave very few offspring next generation. And that actually causes a great decrease in population size, effective population size. So perhaps it's that, but it could also be other factors than that. It's still something that's being debated what causes that. But this is an example of how you use coalescence trees to make inferences about population history. Formally what you do is that you sample a lot of these different trees, and then you use this Felsenstein equation to get a likelihood function that will allow you to estimate changes in population size like this. Of course, then this happened over the past 20 years. This is an old slide now, this 10 years old, but this explosion, next generation sequencing data, explosion of the ability to sequencing data. We moved from looking at just empty DNA or Y chromosome data to looking at whole chromosome. And that had the consequence now that we don't just have one tree we're looking at. We have millions of trees. And why is that? That's because every time you have a recombination event in the history of the population, rather in the history of the sample from the population, then you get a new tree. Each recombination event causes a branch in the tree to be moved from one place to another place. So in the human genome, you don't just have one tree, you have millions of trees. Each position in the genome has its own tree, but there are millions of them. If you walk from left to right along the human genome, you'll encounter a new tree every time there's a recombination event. Okay, so that means that we can't easily use the Felsenstein's equation. We can't easily do that kind of inferences, sort of model-based inferences. And what you have seen in genomics and population genomics over the past 20 years is that there's been a focus then on sort of simple summary statistics. Typically, people calculate just the frequency spectrum, or that do a PCA, or a structure, or a mixture plot. Other things that can be easily calculated and visualized, but not these sort of model-based inference using all the data. When you do that, you do use typically a great reduction of the data, such as the frequency spectrum, the site frequency spectrum, where you can do some of the same things, but at the cost of taking all the genomic data and just reducing that to a collection of allele frequencies, which is a very, very drastic reduction of the information that is in the data. Okay, so a little bit about what is the structure that you get with with recombination and the multiple trees in the genome. What we have here is what's called an ancestral recombination graph. You can think of that as the collection of all these coalescence trees that are all over the genome. So as I talked about, you move from left to right. Every time you see a new recombination event, you get a new tree. So for example, in this example here, we have a coalescence tree here. Then we imagine that there's a recombination event here. Then there's a lineage here that's free to coalesce somewhere else in the tree. In this example, it ended up coalescing up here, making a new node 8. Then as we move from left to right, then maybe there was a recombination event here. This branch got broken up, and you got a new coalescence event up here. So it moved this branch to coalesce up here instead. And that way, you get a new graph structure that is all these coalescence trees, plus the way that they relate to each other, plus the information, for example, that this one differs from this one by only getting rid of this node and then making a new node that becomes node 8. Okay, so that's what an ancestral recombination graph is. You can think of it as just a collection of all of these trees. And actually, the idea of ancestral recombination graphs goes back to Dick Hudson's work back in the 80s, where people have been aware that this is the appropriate structure and the appropriate way to think about the coalescence process when there's recombination. But it's only really the last 10 years that people have made progress on actually using that representation for data inferences. So I want to go back to Felsenstein's equation here one to ultimately be able to use that one. We can't use that one on these ancestral recombination graphs because there just are too many of these genealogies. So, the space of possible ancestral recombination graph is too large. So what people did, as I mentioned, that they have moved on to use all these summary statistics, site frequency spectra, and so on instead. That is at least until this paper was published, and I consider that to be a seminal paper in our field, in that it showed that you can actually really get a genome-wide inferences of ancestral recombination graphs probabilistically. This is a paper out of Adam Sibbel's group. And what he showed was that you can actually take genomic data and then probabilistically sample ancestral recombination graphs. It's slow. You can only do it for a relatively small sample and so on, but it's actually possible to do that. So many of the same things that people did in the 90s and the beginning of the 2000s using Felsenstein equations on mtDNA data or Y chromosome data, we can in fact do the same thing, at least in theory, on ancestral recombination graphs. After that, several other papers have been published, and in fact also some papers before that show methods for estimating ancestral recombination graphs, and can do it for much larger data. So this paper by Leo Speidel and co-authors, for example, published a method called RELATE that showed that you can in fact do it for thousands of genomes. You can estimate ancestral recombination graphs, but it comes at a cost, and we'll talk about that later. You can't quite do the same kind of probabilistic inferences as you can do with the original methods, ARGs as it was called, and there's also cost in terms of accuracy. And other methods later on were also published that scaled this problem up to even larger samples, but again at a cost in terms of accuracy and in terms of statistical interpretation. Okay, so let's talk a little bit about what you can do when you have actually, if you can sample these ARGs. So sampling the ARGs, these ancestral recombination graphs, that is something that in this paper by Adam Sibbel's group, the ARG paper, was shown could be done probabilistically. And well, first you can do that under one model. You can then make inferences by, in some sense, correcting the set of graphs that you get. So typically we will sample ancestral recombination graphs from a standard neutral model. That is a model that assumes constant population size, and no selection, no population structure, and so on. But how do we then use those graphs for making inferences about some of the things we're interested in, which might exactly be changes in population size and population structure, natural selection, and so on? Well, what we can do is we can reweight the samples. So we can put a weight on the samples according to the probability of seeing those ARGs under the standard neutral model and under other models, for example, selection models. And by that, we can use a trick that's known in statistics called importance samplings to do a reweighting that will allow us then to make inferences under these other models. So when first we can sample ARGs under one model, we can use those sampled ARGs, as long as they're sampled under a well-defined distribution, to move into the space of other possible models where we can make inference for those models, again, by looking at the ratio of probabilities of the ARGs under the different models. Okay, so I'm going to show that how we have used that to look at natural selection, and just reminding you a little bit about natural selection, how it works. Imagine we have a population here with individuals in it. At some point in time, we have a new mutation. If selection acts against that mutation, so individuals that carry the mutation, they have a fitness disadvantage, with high probability that mutation will be lost from the population eventually. So we call that negative selection, selection acting against new mutation. Positive selection is the opposite. The new mutation carries a fitness advantage. Individuals that carry that mutation are more likely to leave offspring in the next generation than individuals that do not carry that mutation. So with some probability, eventually everybody will carry that mutation. In the end, that mutation will spread in the population. So this is positive selection. The new mutation is favored by natural selection. And often in evolutionary biology, we are interested in detecting positive selection because that's what's associated with adaptation. We're interested in how organisms have adapted to the environment, have acquired new functionality, a different functionality. We want to relate what's going on at the molecular level to what's going on in the ecology. That's typically what we're interested in, positive selection. So the traces of positive selection, we often see that as what's called a selective sweep. If we imagine here a line here that is a DNA sequence, a haplotype in a population, each star here is a mutation, then at some point in time here in my figure, a new advantageous mutation arises. That's that red one. And as I explained before, if it's advantageous, it confers a fitness advantage to individuals who carry it, then eventually, with some probability, everybody in the population will carry it. But if there's no recombination, it's not just that mutation that will increase in frequency. It will also be the other mutations that will link to it, these two mutations here in my figure. So eventually, these two mutations will also go to fixation, and the other ones was lost. That's a selective sweep. We end up, after the mutation has gone to fixation, with a local reduction in genetic variability, because all mutations have either gone to fixation or been lost. And we're looking for patterns in the data that look something like this. Often it's not a complete sweep. It hasn't gone completely to fixation. We see something intermediate between this, and that's the kind of pattern we're looking for. The idea here is that often when we look for this, we want to look at what's going on in nearby sites and devise various statistics that can do that. But with ancestral recombination graphs, we can get all of the information from all of the sites in the genome about what's going on in one site. We can get the full likelihood function for the selection coefficient, the parameter that governs how much selection is acting on this mutation, by looking at the ancestral recombination graph and basically doing what people did in the 90s and early 2000s with Felsenstein's equation. So we implemented that idea, and this is some work by my former student Aaron Stern, in a program that does two things. It takes in sampled ancestral recombination graphs from, for example, ARGweaver. So get the samples of different ARGs. So for each position in the genome, there's a tree. And that's sort of some hidden layer. It has to numerically integrate over this space of all possible ARGs, and it does that by sampling representative ARGs using ARGweaver or some other ARG sampling method. And then in the next step, it has to connect the ARGs to natural selection at a particular site. And it does that with a hidden marker model, essentially, that models the change in allele frequency back in time. So you model explicitly how the allele frequencies change back in time. And when you have that, then you can predict how the ARGs would look given the allele frequency in a particular locus back in time. And then we can do this trick with importance sampling that I talked about before, because of course the ARGs have not been sampled under any assumption about selection. They've been sampled on a neutral model, but we can reweight, use this importance sampling trick. And in the end of the day, what we get from that is something like this. So we get what we have here to the left is a likelihood function for the selection coefficient. So let's select the selection coefficient. That's a parameter that specifies how the fitness increase you get from having the mutation versus if you don't have the mutation. So it's typically the central parameter we're interested in when we're interested in selection. And these are likelihood functions here. So the higher the value of the function, the more likely that selection coefficient is. And here in the bottom, what we have simulated is where the true selection coefficient is 3 times 10 to the minus 3. And we can see we get an estimate that's very close to the true value. So the red is the true value, and the blue is the inferred value in this example. Here's a case of neutrality. The true value is 0. We get an estimate that's very close to 0. In fact, not significantly different from 0, whereas this value would be significantly different from 0. So we can make now these likelihood ratio test hypothesis about how much selection is acting, and also infer what the selection coefficient was. And in addition to that, because we had this hidden marker model, we also get estimates of what the trajectory of the mutation was back in time. So this is a neutral mutation. The red path is the actual simulated path that when we simulate the mutation, and what is in yellow is the inferred allele frequency back in time from the method. This is a case of where the selection, the true frequencies are in red, and the inferred ones are in yellow. So in this way, we can infer by just looking at the present data what the allele frequency was back in time using the ancestral recombination graph. And then we can do simulations, and we can show that doing this, we get tremendously more power over standard methods for detecting selection. These are these sort of standard ROC curves that try to look at comparing different methods. I'm not really going through it, but in simulations, you can show this actually performs quite well and can lead to a lot of power, particularly when the ARG reconstruction method that is being put into the program works well. Here's an example of that. You might be familiar with lactase, the lactase locus, that if you have a certain allele allows you to digest lactose as adults, have lactase persistence, or avoidance of lactose intolerance. And it's one of the examples where there have been strongest selection, at least in European. Selection has also been in several other populations outside Europe that have adapted to drinking milk as adults. But in Europeans, we see the strongest selection, perhaps any place in the genome in the lactase locus. And what we're looking at here is the allele frequency trajectory inferred from ancient DNA data here, from this Madison and Madison paper. And then the allele frequency trajectory in black that we infer for just the modern data using the ancestral recombination graph. They track sort of reasonably well. They're not perfect. We think part of the discrepancy has to do with that maybe the populations that have been sampled from the ancient populations might not be exactly representative of the ancestors of this modern population, and that could cause some minor discrepancy here. But roughly, we are inferring the same pattern as inferred in Madison and Madison here. Here are some other examples, other loci that have been claimed to be under selection, and what we see for some of the loci, we see strong evidence of selection. For example, the KIT ligand here, we have a likelihood ratio of 30. That's a very small p-value, very strong evidence that there's selection acting on the KIT ligand, and a very strong selection coefficient on 1.6 times 10 to the minus 2. And here another locus in the mutation in the ASAP locus where we really don't see any evidence of selection. Log likelihood ratio of 0.39 and no evidence of selection. So in this way, we can go through the genome and we can quantify how much evidence there is of selection at any point in the genome. And this is also, it doesn't matter if the mutation has gone to fixation, it doesn't have gone to fixation, it's a soft or hard sweep or anything like that. As long as we think of it as selection acting on a specific mutation, then we can ask how much selection is acting on that mutation, what its frequency was in the past, what's the statistical evidences of selection, and so on. Okay, so that's all good and fine, but typically in real data, you don't have, it's very hard to discover the relationship between the genotype and phenotype directly. And in particular, many loci might affect the same phenotype. So if you think about human genetics, we're typically not that interested in just a single locus, because most of the phenotypes we're interested in, they're affected by many loci. They're highly polygenic. There are few examples in humans of heritable traits that are not highly polygenic, but almost everything we are interested in is highly polygenic. So if we're really interested in selection on a trait, we should somehow use the information from all of the SNPs that might affect the phenotype. And we can do that because there's standard statistical methods for doing this in statistical genetics and quantitative genetics models, actually going back to work from Landy in the 70s for how to combine the evidence of how to combine selection from multiple SNPs acting on the same trait. And we have implemented that in a program called PORM that we can show also has quite a lot of evidence, or quite a lot of, power to detect selection compared to previous methods for detecting polygenic selection. And by using the explicit model here, we can then also take into account factors like changing population sizes, and we can, using various traits, also to some degree deal with population structure when we try to get at selection acting on a specific trait. And we implemented this program called PORM, and we have also a new version of that called PORM2 that I might talk about a little bit later. So this is an example of applying this idea to UK Biobank data. We looked at different traits, then we took all the SNPs that affect that trait and applied this method. And I know you can't really see probably very clearly the slide here, but zooming a little bit into the left, these are some of the phenotypes where we found strong evidence of selection from people of British ancestry in the UK Biobank. There are some, for example, a trait that's related to metabolism, to type 2 diabetes in particular, but the trait where we found the strongest evidence of selection is tanning. So what is tanning? That's sort of a self-reported trait. People that are on UK Biobank, they fill out questionnaires, and this is a trait that comes from that. And that's because they don't measure skin pigmentation in UK Biobank, but they have these proxies for it, such as tanning. And it's clearly something that's closely related to skin pigmentation. And so if we look at the UK population, ask in their past history, among the traits we've been looking at, where do we find the strongest selection? We find it associated with skin pigmentation. And many other studies have actually argued something similarly, that there are many loci that have been under selection in Northern European associated with skin pigmentation. Now, what I talked about before was the problem that you might have polygenic traits, that many SNPs affect the same phenotype. But you, of course, also have SNPs that affect multiple phenotypes, your pleiotropy. So in real life, you know, even in simplified forms, the way genetics relate to phenotypes is more like this. You have many loci, and for each locus might contribute to multiple phenotypes. Now, fortunately, we can also address that. There's also standard methods from statistical genetics that we can model these genetic correlations for human data, where we know effect sizes for these different phenotypes. So as long as we have the effect sizes measured for each SNP for the different phenotypes, we can also entertain models like this, where there's correlation among traits, that some traits have a shared genetic basis. For example, this phenotype 1 here is affected by this SNP. Phenotype 2 here is also affected by this SNP. So these two traits are genetically correlated. All right, so genetic correlations, we can take them to account. I'm gonna give an example of that. This is an example from the UK Biobank again, and on the issue about whether there's a selection on European hair color and eye color. So this is quite controversial. We know there's been selection associated with human skin pigmentation in Europeans, and we think we know why. And the story is that you want to have more pigmentation if you're exposed to a lot of UV radiation. But because a lack of exposure to UV radiation can cause vitamin D deficiency, you want to have less skin pigmentation when you are not exposed so much to UV radiation. So the argument goes that in Northern Europe, there's been a lot of selection against having too much skin pigmentation to avoid vitamin D deficiency, and that in other parts of the world, selection could have worked in other ways, that this is sort of trade-off between vitamin D deficiency and the damaging effect of UV that causes different types of selection in different parts of the world, depending on how much you're exposed to UV radiation. But then why does hair color vary? And there have been papers, for example, this paper that's been arguing that it is because of sexual selection. So in some sense, we're humans a bit like peacocks here, that, you know, the sexual selection story in peacocks, where males, they have this massive display here of their feathers that most people, although that's also controversial, but some people at least think is great disadvantages in terms of survival, but it increases its access to females. So there's sexual selection for the trait. So people are arguing that maybe something similar is going on with hair pigmentation in Europeans, that's formed by sexual selection that's been going on. Now, what we can do now is we can deal with these correlated traits in this framework and then ask the question, if you take into account the selection that has been acting on skin pigmentation in Europeans, do we actually then see any evidence of selection on hair color? And the thing is that the evidence almost entirely disappears when we do that. That it seems like the vast majority of selection that affects hair is really selection on skin pigmentation. But because some of the same loci affects both hair pigmentation and skin pigmentation, there's been correlated selection on hair pigmentation. So we think that at least the vast majority of selection that has been on hair pigmentation, on hair color, has nothing really to do with sexual selection, but it's really caused by the selection acting on skin pigmentation. And it's just a byproduct. The change in hair pigmentation in Northern Europeans is simply a byproduct of the selection associated with skin pigmentation. That's at least what these results would suggest, that we can explain this apparent selection on hair pigmentation, on hair color, by selection exclusively on skin pigmentation. All right, so that's one thing that you can use ancestral recombination graphs for. You can extract all the information about selection on the locus by using ancestral recombination graphs. You can do many other things to it. For example, you can look at geographic structure. And this is some work in progress by my graduate student, Andrew Vaughan. And one of the things he's been interested in is population structure and how to use that for looking at population structure. And he makes models then where he have populations on a grid like this. So each node here is now a population, a geographic location, a population that lives locally in an area. And then he looks at gene genealogies, local coalescence trees in the ARG, and try to trace back in time where people might have lived in the past. So here's an example from some European data from where we used ancient DNA in combination with modern DNA, and the samples here, the red samples, that's ancient DNA samples. And then we can take a modern individuals from England. Here we do it for chromosome 1, and then we can ask where did this individual's ancestors live in the past? And here we go back 2,000 years ago. This is probability distribution of possible ancestors. We see sort of have moved over to Northern Europe, and we know that there was this big migration of people from Northern Europe, from Scandinavia, and what today is Friesland and Northern Germany, into England about 500 AD, and this might possibly reflect that. If you go back to in time 8,400 years ago, we get that much of the ancestry lies over here in an area pretty much where Ukraine is today. And what we think this is that there was this famous migration of the Yamnaya people, these steppe people, in the early Bronze Age that came in through to Northern Europe and also spread to much of the rest of the Europe that we know now from ancient DNA. And we know from ancient DNA that many of the ancestors of people in England, in fact, traces back to that migration. And this way, using ARGs, we can, for a particular individual, trace the ancestry of that individual graphically back in time. Yeah. So this is these Yamnayas that I was talking about, an archaeological dig from a Yamnaya burial. All right. So all of this, so there are lots of things we can do with these ancestral recombination graphs, the ARGs, but it all rely on that we can estimate them well. In fact, it really relies on that we can sample from a posterior distribution of them well. So how well do current methods do? So we've been looking at into that quite a lot. So here's an evaluation of how well these methods estimate coalescence times, for example. And there are 3 methods here. ArgWeaver, that's that original method by Adam Sibbel that uses complicated computation to sample posterior ARGs. And then we have relate, that is much faster, but comes at a cost. And here another method, TS-infer, together with TS-date, that is even much faster. This can analyze UK biobank data. It's really remarkable, actually. You can analyze such large sample size, but it definitely comes at a cost. So here you can look at, for example, the mean square error in the estimate of the coalescence time. For ArgWeaver here, it's 0.4. For relate, it's 0.6. And for TS-infer and TS-date, 1.6. So in terms of how well the ARG is estimated, there's definitely a difference between these methods, with this original method being substantially better than the other one, and this TS-infer and TS-date being quite a lot worse than the previous methods. Okay, so my student Yun Deng, who I supervised together with Yun Song at UC Berkeley, he was interested in this problem about making ARG inferences. And the question is, we have methods like this that are highly scalable, but not very accurate. And then we have ArgWeaver that's very accurate, but very scalable. Is it possible to move up in this space, making something that's more scalable, but has the same accuracy as ArgWeaver? And that is basically what his PhD thesis is about, to develop a new method for doing that. And the method is very similar to ArgWeaver in many ways, but he uses several different tricks. And perhaps the biggest innovation is that he decouples the way that you explore the topology of the ARG and you determine the branch lengths. The way ArgWeaver work is that you take a haplotype or a branch in the tree, and then you thread it in where it fits in the ARG. You sort of put it on, figure out where do you coalesce in genome this new leaf node or internal node that you can rethread into the tree. And without getting too technical, what Yun Deng here is doing is he's saying, let's do that first on the topology and then determine what the branch length was afterwards. And it turns out that little trick can speed up the computations by orders of magnitude. It changes the scalability of the inference procedure radically. He also inferred a few other things to get at these branch lengths a little bit better in the inferences. So here's a comparison of the seconds it gets to estimate ARGs using ArgWeaver and this program that's called Singer. It's vastly faster than ArgWeaver. Compared to Relate and TS-Infer, it also has remarkably better performance in terms of its estimation of, for example, allele ages or anything else that you want to compare. Here's another illustration of why it's faster. It's my ArgChain Monte Carlo algorithm, both ArgWeaver and Singer is trying to simulate to get to some stationary distribution. You want to get to where it's in equilibrium. Here's a run with Singer and the same data with ArgWeaver. And you can see the rate of convergence is just much, much slower for ArgWeaver than it is for Singer. Yeah, we zoom in on this little area here to the left. You can see Singer goes in and then very fast it converges in an area like this. And if you keep running the program, it'll keeps going in here. And for ArgWeaver to get to that, it takes us a very long time. So it converts, it's more efficient than MCMC algorithm, it converges much faster. Turns out it actually also provides better estimates. And we're not quite sure, and we've been looking at maybe it's convergence issues in ArgWeaver, maybe it has to do with discretization, but in all our simulations, we tend to perform a bit better in terms of estimation than ArgWeaver. So here's a comparison of Singer, ArgWeaver, Relate, and TS-Infer. And this is the mean squared error, so the lower you are, the better you are. And it performs like ArgWeaver, better than Relate, that performs better than TS-Infer, but it actually also performs better than ArgWeaver. And we see that almost no matter which metric that we're using on this. If we look at more sequences, 300 sequences, we can't use ArgWeaver anymore. It's just too slow to do that. We can look at another method that recently published ArgNeedle, and it also performs better than that. That has a performance sort of in our simulations comparable to Relate. We can also look at the topology, not just the times the most recent common answer, the coalescence times. Here's what we call triplet topology. It's a measure of the accuracy of the inference of topology. And again, Singer performs better than the other methods. So, what really, maybe I won't go through for the sake of time all of these things in this graph, but an important thing is that it can make credible intervals, so measures of statistical confidence that are pretty accurate. And none of the other methods can really do that. And that's important because we can now really use the ARGs we sample for statistical inferences. We can trust, at least to the degree that the model is correct, we can trust the measures of uncertainty that we're given. We can assign probabilities to events. We can say, what's the probability that the coalescence time is larger and smaller than this, and anything else that you might want to ask about an ARG, we can assign accurate probabilities to that. And that's great advantage because now we can really do the kind of thing about detecting selection that I talked about with the project that Aaron Stern has been leading, and the population structure project that Andrew Vaughan has been leading. They all rely on that you have proper calibrated probabilistic ways of sampling these ARGs, and we have that now. So we're quite excited about that. And there's various tests to do to show that it's probably calibrated, that we can do, that exist in sort of the literature of how to do Bayesian inference using MCMC. And we can show that we in fact have well-calibrated probabilities here. Okay, so a little bit, so as few examples of it, what you get out of it, you can, for example, infer here the times the most recent common ancestor locally. This is here for the ABO locus, a locus that's known to have been argued to have trans-species polymorphism. We estimate the coalescence times here to be larger than 6 million years, the times the most recent common ancestor in the ABO locus in humans. This is for the locus that controls the lactase persistence in Europeans. And what we can look at is the branch length diversity. That is the total length of all of the branch lengths, the total tree length, how much genetic variability there is. And you see there's essentially zero genetic variability here. If you look among individuals that carry the allele that confers lactase persistence, so avoidance of lactose intolerance. If you look at the other haplotype, you see a quite different pattern. Okay, and you get far enough away and you get the same pattern in the two haplotypes because you don't see the signal of a selective sweep anymore. Here's the MHC region in some populations from Africa. So you're probably all, I presume, familiar with the MSC region, which has to do with self-recognition and is really important has important function in the immune system. We know there that there's trans-species evolution, that genetic variability within humans is shared between chimpanzees, that we have some very old coalescence times. And what we can do now is that we very precisely can go in and say, where in this region do we have these regions of very deep coalescence times? So we see some peaks, different places of coalescence times, or time to the most recent common ancestor that are older than 20 million years. And that agrees with the fact that we see shared polymorphisms between species in some of these regions. But the new thing here is we don't have to use a sliding window. So for example, in many analyses, several of these peaks are simply averaged out due to sliding window uses. But since we have the ARG, we can ask, what is the time to the most recent common ancestor at any point on the genome without having to average or use a sliding window or anything else like that. And it gives a much more precise picture of the distribution of coalescence time. So we can make we can pinpoint effects, for example, on natural selection, much more precisely, spatially in the genome. Okay, this is another locus, SPECS3, from the, against African variation, 1,000 genomes, where we look at how much variability there is within a population versus between a population. And where for this locus, we see some very specific patterns that are specific to one population that might be worth exploring. So it can be used for this way to explore directly without even formalizing any statistical methods on top of it, try to explore patterns of reduced variability or increased variability in genomes. Okay, so we have worked more on this ways to use this important sampling using selection, and have a new program out, CLUES2, that we think works even better than the older program. It's certainly much faster. For example, see here inferences of the selection coefficient, quite precise. With CLUES2, we can get very precise estimates of the estimates. It's a paper that was just published a couple of months ago here by Andrew Vaughan. And one of the things he also implemented in this paper is that you can look at varying selection through time. So you can ask, does it fit the data better if there was a change in the way selection happened? And that is really important for many questions, because many questions we have about natural selection really has to do with how natural selection correlates with some environmental variable. And often we want to ask, when the environmental variable changed, did selection change? And that's been hard to do previously, but now we have a formal mathematical framework for doing that, where we can do that. We can formally specify models of changes in selection coefficients, and very easily then test how much statistical evidence there is for a change in selection coefficient. And this is an example here from some different loci. MCM6, that's where you have the genetic variation associated with the lactase locus, so the variation associated that determines whether you have lactose persistence, lactase persistence of expression of lactase as an adult. And what we see here is that if we look at the log likelihood and then compare log likelihoods for different models using the AIC criterion, we get strong evidence that there actually been change in selection in the lactase locus, or in the genetic variants that governs lactase expression in adults. And we can go in and get a much more fine-grained estimate then of how the allele frequency changed through time. And what we find now, in contrast to previous results, that when we now allow a model like that, where we allow differences in selection coefficient through time, we get that most of the selection happened in a relatively short period between 3,000 and 4,000 years ago, where there was extremely strong selection. And that a model like that fits the data way better than a model that assumes constant selection. And that fits with the idea of, if you talk to archaeologists, that in Northern Europe, this is a period where farmers relied very heavily on dairy farming. That dairy farming was a very, very, very important component of farmers, and they were before and afterwards they were more diversified. So we think that this that the fact that there's so strong selection in this period has to do with cultural practices and the role of dairy farming in these communities. Okay. So I'll end up with one last example. It's from some ancient genomes. This is actually not from Ancestral Recombination Graphs, but it's the same mathematical machinery where we take alleles through time and follow allele frequencies through time. This is from a paper we published this year in collaboration with Eske Willerslev's group, who studies ancient DNA. And this is samples that his lab had generated all over Europe, and from a period from almost the present down to about 15,000 years ago. We're then interested in looking at where selection has been acting, particularly in Northern Europe. We have lots of samples, for example, from Denmark, where both Eske Willerslav and I am from. And so we use that to try to look at selection through time. And what we can do is we can take this program, this is from clues, and then using these allele frequency trajectories and the information from those about the selection coefficient, we can make a scan through the genome of where we see selection acting. And this is, for example, HLA, this is the MHC region. Here, one of the famous loci that has to do with skin pigmentation. Here we have CM6, the lactase locus. One particular disease we're interested in was multiple sclerosis, and that's because it has a spatial distribution where it's very common in Northwestern Europe. It has a very high prevalence in Northwestern Europe, and it's supposed to be genetic, and we know something about the genetic background of multiple sclerosis. We know many of the genetic variants, including some in the MHC regions, that are associated with multiple sclerosis. And what we'll be able to show in this paper is that some of the major mutations that causes multiple sclerosis or increases the risk of multiple sclerosis actually came in with these Yamnaya people I talked about before. So if we look at the population in Europe, we can think of three major components: original hunter-gatherers that were there, back in the Pleistocene, then Anatolian farmers that come in about 8,000 to 9,000 years ago, and then these steppe people, the Yamnayas, people related to them, that came in about 5,000 to 6,000 years ago. And many of the alleles that causes multiple sclerosis had a relatively high frequency in these Yamnayas. And we can start then speculating about that. Why is the frequency high of some variants that causes that disease? And the typical hypothesis for this is that there has been a change in the environment, in particular that we live in a much cleaner environment today. So these autoimmune diseases like multiple sclerosis are much more likely, we're much more likely to have them because we don't get exposed to natural pathogens as much. So our immune system becomes overreactive and we risk to develop diseases such as multiple sclerosis. That's because our immune system is too overactive. But some of those variants might actually also be beneficial in other environments. If we are exposed to a lot of pathogens, those very same variants that today increase the risk of multiple sclerosis, 5,000 to 6,000 years ago, they might have been beneficial because you lived in a very different environment or exposed to many more pathogens. And you might need a much more active immune system, particularly when you start to live in denser societies, like you had livestock around you, that is hypothesized to have been a great increase in your exposures to pathogens. That might have driven selection that increased the allele frequency of some of these genetic variants that today predispose us to multiple sclerosis. And when we look at that, we actually look at, we can see that these steppe people, they have this higher ancestral risk score of multiple sclerosis. And moreover, we can see there's been very strong selection for the genetic variants associated with multiple sclerosis in the past. So, for example, here this allele frequency trajectory within one of the Caucasus hunter-gatherers, an ancestral group that has reasonable component in the Yamnayas, and we can look at, if you look in the past, that there's been very strong selection for some of the genetic variants that today predisposes for multiple sclerosis, but that apparently were favored in the past. So we think again that the high prevalence in Northwestern Europe of multiple sclerosis might have to do with natural selection acting 5,000, 6,000, 7,000, 8,000 years ago. When people start to live in societies where there was much higher population density, people live together with livestock and so on, more exposure to pathogens, favoring mutation that causes a much more reactive immune system. And then today, when we live in much cleaner environment, that these very same variants now predispose us to multiple sclerosis. All right, and with that, making sure I have some time for some questions, I'm going to end there. I'm going to just thank my collaborators. and especially the people in my group who have been working on this, but particularly Yun Song, Andrew Vaughan, and also Deborah Brandt, who did, the simulations, and Aaron Stern, who's now no longer in my group, who developed the original CLUES version. And then thank you to all of you for listening to me today.